So, yeah, thanks for having me here. This is another user story, because we are currently exchanging user stories, I guess, to highlight a couple of different use cases that have potentially diverse needs for data accessibility, formats, and chunkability and stuff like this. My use case will be connectomics, the reconstruction of firing diagrams from the squishy matter between our ears or the other end. And this will be a very incomplete story of some of the things that I have run into over my life with these data sets. So if you're missing a tool, we can talk about this later. I try to avoid as many as I can during the course of the following 30 minutes. So what is connectomics? They're trying to, so there's a couple of different opinions. There's an older school opinion of that. Working with MRI and stuff like this, they talk about how brain regions are connected with each other. What I want to talk about here is the real stuff. Getting so high resolution data that you can actually see every individual neuron, every synapse that people are trying to recover from these data sets, wiring diagrams. So how does this look like in practice? And here's an example of this for the Drosophila brain. On the data set that Mary was speaking about, this is a whole data set, you know, and the brain. What you see here is the head of a fruit fly, imaged by a colleague in Jena, Germany. Has its two eyes, antenna. This is the mouth part. The esophagus goes straight through the brain. I don't know, thought that was a good design. And we will now slice through this head. And then we see the brain that sits nicely mounted between the two eyes, which is clever. And then we will see how this EM data set slices through this brain as we're zooming in. And then following that, we will zoom into the EM data set. And if that weren't compressed by Google, so dramatically, you would see some stuff. I'll stop this here. So here we see, you see the mouse. This is the synapse. 3 synapses and some postsynaptic cells. All of those things that are surrounded by black lines are individual neurons. So it's basically a terminal full of cable, and you have to wire that cable. So that's all right. So connectomics is not. It's not one task, but it requires acquiring these images, processing them, analyzing them, manually proofreading them, providing them visualization, and so on. So I've tried to enumerate a couple of things here to cover all those use cases. Starts with image acquisition. You have to get this stuff somehow from a microscope onto hard drive. Then we have to do a lot of stuff with alignment because it's electron microscopy. So you either scan this or you do transmission EM. You have to build mosaics from overlapping tiles, and you have to then keep your section alignment because the individual sections are all deformed. Sometimes the image the volumes, and you have to stitch the volumes together. Okay? And you have to do some intensity correction that people can actually look at this, or computers. And then eventually you want to reconstruct all these neurites from those data sets. You want to detect all the synapses, and you want as much of this be done by computers. And eventually you have to put some people on top of this. It's all many mistakes. Okay. So this, for me, this started with a tool. well, first about microscopy. I'm not a microscopist, but we so this will be the shortest part. When data comes from microscopes, there is a diversity of conventional and specialized data formats, as you all know. Microscopy vendors do things that are appropriate and easy to handle for particular imaging pipelines. So if you have a scanning here, for example, you want to have a file format that allows you to stream out that scanned data very easily. Sometimes microscopy vendors convert this into file formats that are a little bit more user-friendly, like this MRC format from the MRC LFB. which inputs some data and some export you nicely image processing tool handle all files from my in our institute, building customized hardware and to export things as plain binary streams with some metadata headers in front. And if the metadata header in the front gets too small, they put some metadata in the back, so it's a little bit of a mess. So stuff comes out of a microscope in a way that the microscope likes, right? So with these image data sets, you then have to perform alignment and segmentation to get something out of this. And my experience with this in this space started with a tool developed by Albert Cardona, and then I contributed lots of alignment tools to this tool. It's called Tracking. That's a ImageJ plugin, but it is actually much larger than ImageJ because it contains but it includes all the libraries that are available in the world. Provides tons of tools, has its own completely independent UI. It's right-click based because that was apparently convenient. And tracking is also the reason why PG exists, because it was very complicated to install TrackM2 into an existing ImageJ pipeline, and they were looking for an easy way to install TrackM2 with that. It actually worked. And now it includes TrackM2 and you can still use it. So TrackM2 has a couple of features. It aligns things. It allows you to manually correct those alignments. It allows you to do some intensity correction, interacts with Fiji and lots of plugins in there. And it allows you to draw profiles of objects in these large 3D data sets, and it allows you to skeleton trace through these data sets. Right? So it can do it all. In terms of data storage, it does what it can. It supports all the images as supported by ImageJ. These images are referenced by a giant XML file that references all these individual tiles and assigns a transformation to the tiles. And then it stores the annotation data that you apply on top of this into the same XML file. So you can likely imagine that if you have a very large data set of like a few hundred or thousands of sections and a few hundreds or thousands of tiles per section, this becomes a very long XML file. And so the loading time for this XML file ended up in the range of hours. It is sometimes difficult. That is not made simpler by the fact that you improve the alignment tools and you have more and more complex alignment transformations in there. So it started with just affine transforms. It's like, okay, 6 numbers per tile is like an easy thing to do. And then we started more deformable things, and we used Jordan Bogovic's interpolations and moving these squares and all sorts of other gimmicks. And then we ended up storing these transformations into the XML files. And if you store numbers unpacked, they become very long. And so we could play dirty tricks like doing binary encoded base 64 stuff and cramming that into text. And it was a little bit hard. Okay, but the tool actually works. And this is how it looks like. You have the fire called a section series. You have lots of tiles that you can manage these tiles and video glasses. Okay. We later transferred this, the capabilities for alignment of this tool into a tool that started with the weird name render. Only because it started as a command line tool that was called render that stuff, which made a lot of sense. And then it turned into a whole database that converts what TrackM2 can do for alignment, like storing all the transformations and stuff like that, into a database and reference this database more effectively, so you can scale this arbitrarily. And this tool was then used to generate this 5D data set that you've seen in the beginning. Render doesn't take care of segmentation, but only for alignment purposes. As I said, it makes it more efficient to store these transformations. It still references original tiles, and it makes one step forward. It generates bitmap representation of original image files that it can then use to more reduce resolution levels. It exports data to HHMI, which we come to next, and find which we are better about, and to the next. Okay, what's HHMI? HHMI is a web tool to support the skeleton tracing initially that TrackM started with. We started building this in 2009, so it's a long time ago. And then colleagues at Janelia and it so it started just as a visualization tool, and then our Cardona Stephan Janelia implemented all the skeleton tracing tools and graph analysis. It became this massive workbench that supports everything that is skeleton-based connectomics analysis. Here's a screenshot of CatMate, the smallest part with viewer. That's the stuff that I wrote at some point. Then it's all the rest, the super complex, and includes the 3D viewer and then all sorts of stuff. One thing that CatMate can do is it can display ortho slices. We have here a small video for this. In order to do ortho slices, you need alternative exports of data sets. So it exports them into little JPEG files like Google Maps viewers or similar tools. And you can export several axis permutations, relate them by different scale factors. And then you have this nice little tiled image window manager. And you can basically have arbitrary-oriented views in data set by doing redundant exports. Well, those were skeleton tracing tools, but of course, it would be nicer to have actual volumetric segmentation of these structures. And one of the things that we learned over time is that we don't have enough training data to actually use proper deep learning tools. And so we were interested in generating more and more comprehensive ground truth data. A lot of groups that were doing similar things. Our ground truth data set at that point in time was eventually 5 times as big as all the previously existing ground truth data sets together. We're proud of the way how we created this. Is we built a tool that built on top of another tool that we had developed before for completely different reasons. The big data viewer that you may have all seen in Fiji. That is based on ImageNet 2, and it can arbitrarily slice into very large data sets, load small chunks of data at multiple resolutions. We decided at this point to use HDF5 because it's a really beautiful hierarchical file format. You can put a lot of structure in these data sets. You can associate metadata to data, and you have everything in a big box. Little small chunks from these HDF5 data sets. And we built a tool that allows us to very, very quickly paint 3D stuff into relatively large volumes from these HDF5 data sets. So you see how this tool works. So you can click around, you can reslice, you can change colors, overlay, brush tools, and you can paint in these data sets. That was fun and great. But at some point, we learned that HDF5 is not the final answer for really quick data sets. We're going to come to that later. So meanwhile, at Janelia, the FlyM team developed proofing equals for SEM data sets. So what you've seen so far are like TM data sets. At Janelia, we're doing kind of 2 things like TM, non-isotropic EM data sets, and isotropic FIP SEM data sets. And for these FIP SEM data sets, the FlyM team had collaborated with Google to generate automatic segmentations. Google had trained a segmentation method that they called clustering networks, which generated very high quality segmentations, but you still need proofreading. And the FlyM team built around this setup a set of tools that consisted of a data access API divot and several different tools to do proofreading operations. The interesting thing about this data processing API is that, first of all, it is a chunked access API to data sets. It implemented version control over these data sets, which is an interesting idea. So you can modify the data in this data set. It only stores up the execute chunks that have been affected like this. You can generate arbitrary snapshots for these data sets, and it's relatively transparent to the user how they are generated. Divot also implements a subpub API to synchronize data sets and different representations between them, right? So if you're interested in all of this, you have to read all those papers. This tool, the FlyM team by now has generated several fabulous connectomes of the central food brain, the optic lobe, the ventral nerve cord. This is a rendering of the ventral nerve cord, part of the toolbox. They generated is also a tool that helps render these beautiful movies. They call this new bit. Generates Blender exports and uses Blender to make renderings from their reconstructions. Next one. Meanwhile, in the rest of the world, other people are also doing connectomics. And some remarkable databases that I wanted to mention here were the Open Connectome project, the follow-up project, BossDB, and a project from Vim Cloud. All these data stores try to do kind of the same thing. They want to provide you a web-based API, some REST thing that can ship you chunk data or small views in chunk data sets. So you can use it in arbitrary tools like HHMI, like these usual tools and other applications. And so the idea that all these tools basically share is that they extract chunks from some sort of native storage, and they export several of the various native. Okay. All of those tools don't address image acquisition, alignment, or anything, but they're focusing strictly on segmentation, visualization. With that, we come to the identify. So hearing about those tools is one common thing is that we see among all of them is that we have very large data sets and we want to all people. We were able to do this with HDF5. Yeah, that's great. But it doesn't really help you when you do things over the web because there's no dedicated read and write access into small chunks of HTML5 through the web itself. And so we had actually one scenario. We did a stitching pipeline for Eric Benzig's lattice HHMI Cosmos data sets, and they generated data set like 30 teravoxels. And we wanted to export this, and we were like, oh yay, let's put this into an HDF5 file. And it took like 2 weeks to export it. And then we thought like, but that's stupid. And what the HDF5 file is basically doing is like a directory tree, and we're using a file system in a file, and there's really good parallel file systems, so why shouldn't we use a file system? So let's write this super simple, dirty hack that is like storing individual chunks as individual files, and we put JSON on top of this. We have that data, and basically everything in HDF5 class thing. So we wrote this in a week or so. It's relatively short, like the first file version. And then we exported this, and it took like half an hour on a computer cluster to export this because parallel file systems actually work. It's really cool. And so that is how N5 started. And we browsed the internet for the right terminology and figured out all that's possible. And then I have already a thousand files. And it's like, hey, we invented the same thing. And it's like cool. And since then we're talking about this in the deal. One outcome of this is that the N5 API that we wrote is basically not just a format, but it's an API for all sorts of chunk-based formats. So currently you can use the N5 API to play N5, XOR. You can do this in Google Cloud, on local drives. You can do some other mappings. It can also read and write HDF5, and you don't see this as a front-end user of the library itself. Is basically everything's the same. And all of this is available in DG. So I'll institute people if you want to export something that uses all your cores in parallel, and the data set itself fits in main memory. So if it's 50 gigabytes, you can load it on a MacBook. Then you can split this into DAR or PNGFF with the DG plugin. Maybe that's and it's scriptable. Right? Okay, so. Yeah, this is how N5 worked. We'll get to an end here. We use N5 in registration pipelines for these connectomics pipelines because we had to store super large information fields. And we had to load this data. So this data set here, for example, is like 35 teravoxels in size. So you can't load this in one piece, and you have to deform all those pieces interactively. And you want resolution. Well, resolution so you can zoom out. cement this to trace all the neural fibers. But you see right here, in the middle of the screen here, this line is actually where the what is the stitch, where it fits, also screen. The best library to store these multi-dimensional arrays very abundantly, I think, is TensorStore. It doesn't take care for the grouping and the metadata, but it supports N5, ZAR, and several other backends and neural network. Sorry. Okay. So there are tools to use those backends. Number one is WebNosos. That's for tracing and signal handling segmentation. Developed by Scalable Minds in Germany. Little screencast that shows you how WebNosos can be used in those very large data sets. It supports ZAR and 5 and some else based on. Another tool that we developed for somewhat similar but also different annotation tools to Pantera. Pantera, you can arbitrarily reslice and use singular magnetic segmentation to paint into extremely large volumes. We use N5 or HHMI. Data backends, it works for cloud store or local archives anyway. Web-based tool, you've seen it before. Neuromancer does more or less similar things, but doesn't allow you to paint at this point. But people have implemented extremely large and powerful implementation pipelines almost at the end. To proofread some of the largest data sets in the world. And this works all in collaboration with a set of tools and services that is called CAVE. Okay, so this is stuff for discussion. Some lessons learned is microscopists want to stream. Screens are 2D, but reslicing is 3D random access. That makes a difference. If you don't want 3D random access, 2D random access is cool. If we're training neural networks and we want to train 3D neural networks, we want 3D random access. If you want to train 2D networks and you want 2D random access, then the 3D blocking is actually super annoying because you're loading too much stuff all the time. It's terrible. Smaller 3D chunks are great for most things, except for hexaline 2D random access. The line average tracing the way all that information, so I'm missing this, and don't worry, it's going to talk about this more and stuff more. As you can see, all we done in naturalizations, like scale levels and deformations make things a lot faster than very small. I haven't talked about time at all. And then there's a couple other things. Scaling is hard if you're trying to build a database, use a database. And things feel like a parallel task and don't reinvent it. It's really hard stuff and people working on those things forever. Most commercial ones are going to Google and Microsoft. I haven't seen these happening. HHMI, in my opinion, is a fantastic file format for almost absolutely everything if you don't have to host small things through web APIs or if you exceed the teravoxel size, because then it gets a little bit unwieldy. With VG, you can store OME and JPEG and HTML or event times, but on VG you can load it. Sorry for that, but it's not good. And that I think I want to end this thing here in discussion. I have a question. Music. Nice.
Yeah, going back to your last slide, you say training 2D, 3D. So did you have to do twice the conversion in order to adjust to the various tool what you wanted to track as part of the use case? Sometimes no.
For most things, I think doing relatively moderate size 3D cubes is the thing you want to do. Because for visualization, for example, you're usually not randomly jumping around, but you're scrolling through things left and right and so on. So you have a little bit of spatial context, so that's useful. Where it actually hurts to have these really blocking reconstructions is that if you're training something like cell phones, like it can really and you actually want to train it with random cutouts from your training data set and stuff, then having 3D blocks means loading block size that much data as you actually want. And that can actually work. So in those cases, it is usually very practical to make a small re-export. Yeah. Well, the training pipeline, then it's just done.
Do you believe that in a few years we can access, like, if it will be like a video game that we can access really easy, and we can simulate the experiments before it happens?
So I don't know if I get the question right, but so there are definitely so most of these applications that I showed you are made like video games. So you can drag and drop things and you can move them, you can paint and stuff like this. Particularly WebNosos has a tracing mode where you fly through the neurons and it tries to render them like you. I found it not so useful, but it's fun. Did I get the gist of the question, or is that all those data sets are super accessible? They're all openly available, and you can use these tools to browse them and to work with them. It's very easy.
Yeah, because for me the important part is that we can save money and time, simulated experiment, and then applying if it's going to happen in real life based on what you're doing.
This is built to simulate an experiment. Or at least I don't see it.
Yeah, but maybe in a few years.
So some people are writing simulators to make data that looks like connectomics data to contain ground truths based on out, because then you have ground truth and you don't have to like manually label stuff. But it's probably not exactly what you were asking for.
Okay, don't worry, it's okay.
Yeah,
Thank you.
I guess you.
Thank you. And I think Alex will lead us into flash talks.
Yeah. In a minute.
Oh, okay. I guess then should we open questions for any of the last speakers? If anyone has anything to add? Or is this what's your advice?
Get everybody up here and make sure they have everything done.
And if I could just add, Eric, while we're sitting here pondering, tomorrow I'll be asking, I think it's tomorrow, what does everyone want to add to OME czar at least? What's the take home of the big projects? How are we going to prioritize and how are we going to build these things? And so mull that over. I mean, we've gotten a couple of to-do lists. There's the Google Doc that's in the Slack. Maybe someone can, write down anything you're thinking, but this gets back to the metadata question from to Dan, but I think it's also for Graham and Sean, which is more of, it's our primary outcome from the meeting really is finding those massive things that are too hard for any one of us to do. And so think about it, and I'll be asking again tomorrow.
Each one's gonna do it on their own. Okay. Dominic, you're up first.
Can I suggest that we get like 2 more people, the next 2 lined up already?
Dominic, Lior, and Conrad. You're the first 3. You're looking for the order. It has been updated in the online program. And if you look at the screen there, you'll find your presentation. Okay. When it downloads, you should be on.
Yeah.
Good.
Yeah, hello, I'm Dominique. I'm from Ibrahim Lab from University of Virginia. I also deciphering the mouse intestine, in which I will be talking about current optimization of intravitreal microscopy of mice intestine with the help of very nice confocal spinning disk system. And when I started this, I didn't have anything apart from this. With a lot of patience, 3D printers, hot glue gun, and access to Amazon, I used a couple of concepts. First one was something like this, where I printed the stage and additional holder for the tissue to stabilize in all axes. Then I went to more sophisticated structures which are isolating the intestine from the body, so there is no movement from breathing or heartbeat. And so what are the results? One of the coolest videos that I managed to record is wave beta neutrophil inside blood vessel of intestine, which looks like it's walking. So this is definitely one of the coolest things. Apart from it, when I was playing with GFP tagged mice, GFP was tagged to some junctional protein. I noticed with lower magnification that I look at not only crypts, but also slices of villi in living tissue, which is extremely cool because whenever you think about structure of villi and crypts,
30 seconds.
So looking differently, you can look at 3D architecture of how crypts and villi. And I've calculated plenty of symmetry and stuff. And the last thing is going 3D in time. So this is an example of first of very few 4D recordings of the tips of villi in mice intestine.
5 seconds.
Visualized in Siglass, which is very cool software. And I actually brought VR headset with me with posters. So yeah, come and see that.